uh, most of the atypical bacteria, I have seen that they tend to be in the order of gram negative bacteria, mostly aerobes. Of course, there are exceptions, and I can classify them into majorly two types. One which are purely intracellular, one which like may or may not be intracellular, do have some extracellular component. Intracellular ones can be again of two types. See, how does a bacteria get inside cell? This is a cell, this is a bacteria. How will this get inside the cell? It will try to get in like this, which means the membrane will form like this. This guy will travel down over here. This will close over it, and therefore forming a vacuole inside something called cytoplasm. There are bacteria which remain over here for the rest of their life, remain over here. There are some bacteria which tend to come out and remain for the rest of their lives in the cytoplasm. Why is this important? Because knowing this is important because aminoglycosides tend to be extracellular, but there is an exception to aminoglycoside. Cells have a tendency to take up aminoglycoside just like they take up bacteria. Now, this is vacuole, this is phagosome, like lysosome rather. Lysosome can get inside and destroy it. There can be two ways in which aminoglycoside, which is extracellular, can infect intracellular affect intracellular bacteria. If a bacteria is inside vacuole and at some point of the time get out into cytoplasm, it has been seen that just like this aminoglycoside, which is also taken up inside vacuole, if I increase the concentration enough, it has been seen to burst out and come into the cytoplasm, and therefore there it can kill this bacteria. So this is one of the ways in which bacteria can be killed intracellularly by a predominantly extracellular aminoglycoside. Another mechanism can be if a bacteria is inside vacuole and aminoglycoside is also inside the vacuole, but the bacteria is thriving by decreasing the acidity. Inside a vacuole, the lysosome can fuse and raise acidity inside, that is lower the pH. For the bacteria to survive, it can decrease the acidity and survive, but decreasing such an acidity will also make sure that aminoglycoside is safe, because this acidity not only kills bacteria but it also destroys aminoglycoside. So if this bacteria destroys the intravacuolar acidity, then not only the bacillus survives but aminoglycoside also survives, ensuring that aminoglycoside kills the bacteria. This is one of the ways in which it can kill. And there is a way in which it can't kill, that is for Coxiella. So Coxiella gets inside cell, but it is such a hardy bacteria that even if there is acidity, it still survives. So it does not survive by decreasing acidity, it survives by tolerating the acidity, and therefore aminoglycoside, which is present over here, is destroyed. Anyways, main thing is, so there are two ways in which aminoglycoside can affect intracellular bacteria, because otherwise aminoglycoside is completely extracellular. Those two ways are that if we increase the concentration enough, aminoglycoside can burst out into the cytoplasm, killing the cytoplasmic ones. Or otherwise, if inside the vacuole the bacteria decreases the acidity, then aminoglycoside is safe and ready to kill the bacteria. So intracellular can be two types, vacuolar and cytoplasmic. Extracellular can be of two types, which has a heavy predominance of abscess and which generally does not tend to form abscess. It may, but usually does not. Like for example, over here is Bacteroides fragilis. Now this Bacteroides fragilis, why does this have such a high tendency to form abscess? Because the polysaccharide capsule is zwitter ionic, which promotes abscess formation. Anyways, getting over here. Now these vacuole part can be divided into two ways. One, like, point is, these intracellular bacteria as they are intracellular, they cannot survive in the usual blood culture. So to make sure that they grow, we need to add a lot of nutrients. In other words, they are all fastidious. So to grow them, we need nutrients. That nutrient can be supplied in two ways. First, blood agar. Second, yeast extract. The blood agar ones start with B. Bartonella and Brucella. The yeast extract are two, Mycoplasma and Legionella. There is a third class which cannot be grown anywhere. For the vacuolar part, this is like it needs live cell. It is Chlamydia. And for the extracellular wala arm, this similar live cell needing thing which cannot be grown anywhere is Spirillum minus. Now, what are the things that I need to add in blood agar part? The blood agar part needs CCSS, cis, serum, CO2, and lastly soya. So soya can be in the form of trypticase or in the form of BHI. Some additional factors, like this Brucella has some additional factors. I will tell you what they are. So first, Brucella. It is a gram negative aerobic, as we saw, intracellular, like mostly intracellular, intravacuolar type of bacteria. Brucella, we get it from either handling cattle or drinking milk. Since one of the ways is drinking milk, so like if we drink milk from our mouth, from the mouth it goes to stomach has acidity, so Brucella needs to have something to combat that acidity. Needs to have something to combat that acidity, and therefore Brucella is urease positive. First point. Secondly, cattle. There is a Noman classification which says that the Brucella of cow, sheep or goats or camel, dog, pig are different. Cow is called Brucella abortus. Sheep, goat, camel starts with mel. Dog obviously canis. Pig Suis, and there's last something newt. not important. Now they have sometimes asked about antigens in the first and second one. Easy to remember. Antigen of Brucella abortus is A antigen. Antigen of the sheep, goat or camel part, which is Brucella mel, is M antigen. Easy. Now point is, what does it do in cow? Basically in cow it causes placentitis leading to repeated abortion. If something is attracted to placenta, it usually needs sugar. Therefore, here along with this we need to add a sugar, which is usually dextrose. Which is usually sugar. Also, why does brucella tend to affect placenta? I just told you, because of sugar. Which sugar? That is erythritol. So this combination can be found in something called Castaneda biphasic media. Now it should have been very good, but there are chances of getting culture negative, firstly, and secondly, which is the more and very important cause, it takes many weeks to grow and therefore isn't commercially used. So next we had what did we have? Backtech. Backtech grows in 1 week, so kind of okay. Interestingly, we had NART. It gives in a very less time, but it is very costly, so not used. So none of them are practically used. The only thing that is practically used is serology. Now, thankfully, brucella develops a lot of antibody, and therefore these antibodies can be put to good use. The serology can be checked in cow or in human. This will be used by vet, this will be used by physicians. So vets can use 3 important tests: whey agglutination, milk ring test, or rose Bengal cart test. Human can use something called a standard agglutination test, but the issue with that is it gives IgM and IgG undifferentiated together, which is practically useless. So one way to solve this issue is to add 2 mercaptoethanol. Then the mercaptoethanol maar deta hai IgM, so left with only IgG. That is one of the way. Or we can check IgM and IgG together, but like together as in, like the problem over here was that it gave the result together. That is not a problem. It did not differentiate between them. So we can have the result together, but if we can differentiate, like if we can keep them differentiated, that would be good. That is done by ELISA or CFT, which gives basically 2 results, one for M and one for G. This one did not differentiate it like that, so we had difficulty interpreting. Or we can use simply IgG with direct Coombs test. Another problem with standard agglutination test is that it can often be false negative due to prozone phenomena. In fact, this tendency for Brucella to cause Ig, like to make Ig, is so high that sometimes antibody exceeds Ig antigen, which leads to prozone phenomena, leading to false negative. So to prevent this, just like to prevent M and G undifferentiated together, we added 2Me. To prevent prozone phenomena, we check at a dilution of not 1:1 but rather 1:160. So therefore, if it is more than 1:160, then it is positive. Now a few facts. Number 1, Brucella has capsule, but it's useless. So no antiphagocytosis and Khellang negative. Secondly, incubation period is 1 month. Some virulence factors are binders named BUR and BVR. Another point, usually 45 days treatment with doxycycline is recommended, along with, for the reason I mentioned previously, there's also a 14-day treatment with intramuscular aminoglycoside. Usually it is seen that for zoonotics, streptomycin works best, but oftentimes streptomycin is not available. In that case, the second best is gentamicin. So 14-day treatment of streptomycin, or a 45-day treatment of doxy plus a 40-day minimum of rifampicin. In case there is neurobrucellosis, I have to add a third drug, which is zone. Okay, now usually features of Brucella are often kind of analogous to features of Bartonella. What are the features of Bartonella? Specifically Hensila and Quintana. We are not considering Bacilliformis. So Hensila is where the cat bites, papule and erythema, followed by obviously a regional lymphadenopathy. Now, if the person is immunocompetent, nothing to worry, it stops there. In case it is immunosuppressed, then three things happen. The three things in order are the first three letters of Hensila, which is H, E and N. So H, E and N. H for hepatosplenomegaly, which is a non-caseating, because it is like not a TB, non-caseating granuloma, and generally granulomas which are due to intracellular bacteria, they're usually microgranulomas. Okay. E for eye, which is conjunctivitis, and for neurobartonellosis, but very rare. This was asked. Achha. For Quintana, we have 5-day fever and trench TBitis. So, comparing Bartonella with Brucella, there is no counterpart of papule and erythema. There is regional lymphadenopathy. There is hepatosplenomegaly. Since it is not like it is due to granuloma. Since it is not TB, it is non-caseating. Since it is intracellular, the granuloma is microgranuloma. E does not stand for I. E here stands for epididymoorchitis in male versus placentitis of female, which leads to abortion basically. And neurobrucellosis very common. We have a 5-day fever. Here we have undulating fever. We have trench tibiitis. Over here we have sacroiliitis and bone marrow depression. Now Bartonella. Bartonella, not a difficult topic actually, already done. This is the prerequisite. It is a gram-negative arrow, intracellular, intravascular. Drug of choice is again doxy. More than, this is first line, more than macrolide. Second line is fluoroquinolone. And the next is features only. So Bartonella can be due to 3 things: Henzile, Quintana and Bacilliformis. This is due to cat. This is due to body louse. This is due to sand fly. My cat scratches me. Where he has scratched, the area develops papule and redness erythema. The nearby lymph node gets swollen, but that is it. If the person is immunocompetent, it will stop there. If the person is immunodepressed, then 3 more things will happen, which are the first 3 letters of Henzile. H-E-N. H-E-N. N for neurobartonellosis, which is rare because it was asked once. E for eye, which is conjunctivitis. H for hepatosplenomegaly. Y granuloma. Not TB, so non-caseating. Intracellular, so micro. Sure. Okay. Quintana 2 features: 5-day fever and trench tibiitis. There's no tibiitis. I've written this to know that there is shin pain. Sandfly. Sandfly actually transmits a set of 5 diseases: 2 non-viral and 3 viral. Sandfly is called phlebotomos. Interestingly, all the viral stuff by sandfly have man as accidental dead end host. Usually I've seen that whenever a viral thing is being accidental, like is entering accidental dead end host, It usually resulting in organ damage more than blood damage, and usually that end organ is encephalitis. So the virus, there are three viruses: Chandipura virus, Toscana virus, and Phlebovirus, which is a part of Bunia virus. Okay. And in the non-viral part, we can either have a hepatosplenomegaly, then a skin issue, or the reverse in every way. So reverse: skin issue followed by hepatosplenomegaly. So this is hemolytic anemia resulting in hepatosplenomegaly followed by warts or initial ulcer result, and later a severe hepatosplenomegaly. This one is leishmania. This one is this one.